Thank you, Carlos, for inviting me to present our work in this really terrific virtual symposium on acute flaccid myelitis. I think this is a really important topic, and I'm happy to participate. The focus of my talk today is going to be on enterovirus D68, which obviously has a very important role in acute flaccid myelitis. D68 is a member of the Cornaviridae family of non-enveloped positive single-stranded RNA viruses. Its translation product is a single polyprotein that's cleaved into mature structural and non-structural proteins by viral proteases. And typically D68 was found to cause relatively mild respiratory infections. But there was an outbreak of severe D68 disease in 2014 that coincided with clusters of acute flaccid myelitis, suggesting that there might be a causal link between D68 infection and AFM development. And then in 2016 and 2018, we had kind of similar outbreaks of D68 and AFM. And so that led us to consider the possibility that perhaps there are different D68 lineages that have emerged that exhibit different neurovirulence or different neurotropism characteristics that may be contributing to AFM development. And so what we wanted to do is to use genome sequence and other kinds of data to first determine if the virus is evolving due to kind of diversifying positive selection, and to determine if the drivers of diversifying selection might include the evasion of adaptive immunity. And then finally, is there some kind of a relationship between the drivers of virus evolution and changes in the virulence characteristics that somehow might be driving the virus to cause these more severe neurological diseases. So we have been using a database resource that my group has developed through funding from the National Institutes of Health called the Virus Pathogen Resource. And really the goal of VIPER is to provide kind of a one-stop shop of data about human pathogenic viruses, that includes not only the genome sequence data, but also protein structure information about immune responses and immune epitopes in viral proteins and other kinds of related data. It's a free open access database that anybody can use, and it has both data as well as a suite of analysis tools to further explore the data. So currently there are actually close to 5,000 genome sequences for D68 from over 4,000 different strains available in the resource. There are 14 protein structures, mostly of the capsid and the 3C protease, that are derived from the protein data bank. And there currently is kind of limited information about immune epitopes that we have derived, that we've kind of captured from the immune epitope database. But it turns out that those are actually quite useful in trying to interpret how the virus is evolving. So we use phylogenetic analysis to help us really understand the evolutionary relationships between D68 and related viruses and how D68 is evolving. And this is just an example of a phylogenetic tree of the major capsid protein for enterovirus VP1 that gives you a sense for the relationship between different species and subtypes of enteroviruses. And so what you can see here is that there are actually 4 major enterovirus species that infect humans, enterovirus A, B, C, and D. And these enterovirus species are related to rhinoviruses. There are 3 different rhinovirus species in green that also infect humans. And you can see that these different enteroviruses include Coxsackie viruses, ecoviruses, and also poliovirus, which is part of the enterovirus C species. And you can see that, although all of these viruses share a similar genome organization, they actually are quite different from each other from the sequence perspective. So what we wanted to do is to try to explore how D68 has been evolving over time. And this is a phylogenetic tree of the VP1 capsid protein for about 1,600 enterovirus sequences. And at this high level, you can see, based on the branching structure of the phylogenetic tree, that there are these 6 major clades of enteroviruses: A, B1, B2, B3, C, and D. Now, the first isolate of D68 occurred in 1962. There were very few sequences that were kind of obtained after the initial isolation. But then starting in around 1990, we saw the prevalence of D68 infections increasing, and we started to see more and more sequences available for evaluation. So now here what I've done is I've taken the phylogenetic tree and I've color-coded it based on the geographic region from which the virus was isolated, based on continent here. And what you can see is that for the most part, there is broad distribution of these different D68 clades throughout the world, with the exception of clade B2, which seems to be predominantly located in Europe in blue, and then clade D, which we find in Asia and in Europe. But for the most part, there's very broad distribution of these different enterovirus clades. So on the next slide here, what I've done is I've color-coded the same tree in kind of a gradient of colors based on the year of isolation, starting with the earliest isolates in red, and then the latest isolates in this bright green. And what you can see is that the branching structure of the tree kind of closely follows the temporal pattern of isolation. And you can see that now, what's circulating currently, from the 2018 sequences in bright green here, are predominantly isolates from the B3 clade as well as the D clade. Now, on the right-hand side of the slide, what I've done is I've color-coded the same tree based on isolates that are derived from patients with acute flaccid myelitis. And what you can see there is that the vast majority of isolates from AFM cases are coming from either the B1 or the B3 clade. So now here, what I'm trying to do is give you a sense for how the virus is evolving from year to year and which clades are kind of becoming predominant. So the AFM isolates are still color-coded in this kind of pink color, and isolates for each individual year are now color-coded in green. So you can see that, in 2011, we have the emergence of these B clade isolates with these green isolates very close to the root of these branches in the phylogenetic tree. In 2012, we see kind of isolates from B1, B2, A, and D. In 2013, we see kind of the emergence of the initial B3 lineage, as well as the first isolate from an AFM patient. And then in 2014, you see that the B1 clade now has become the predominant clade that was circulating in 2014, with several isolates marked with these blue arrows coming from AFM cases. And so this was this initial large outbreak of severe disease that coincided with these clusters of AFM cases that seemed to be coming predominantly from this B1 clade. And then as we move forward, 2015 was a relatively quiet year. 2016 was another kind of large outbreak year. And now what we're seeing is that the B1 clade has been almost completely replaced by the B3 clade from these isolates. And virtually all of the AFM cases are coming from the B3 clade. Again, 2017 is a quiet year. In 2018, really we're only seeing B3 and D clade isolates circulating in 2018, and all of the AFM cases are coming from the B3 clade. And so this led us to ask the question of, how, what is really driving the evolution of the virus? And just to make the point that when we talk about evolution, it's really a combination of mutations that occur kind of at random within the genome of the virus that then experience different kinds of selective pressures. And so the evolutionary rate really reflects this combination of mutation and selection, and it's related to the mutation rate of the virus, but it's not equivalent. Now, the two main drivers of diversifying selection, and this is a really important key concept. The only way that you get positive selection of viruses is that it has to increase the fitness of the virus in some way. And the two things that increase fitness are an increase in transmissibility of the virus so that it becomes more efficient of human-to-human transmission, and substitutions that allow the virus to escape from herd immunity that's developed in the population. Now, the question is, what about virulence? Is there any way that virulence would be positively selected? And in fact, that's probably not the case. In fact, if you have highly virulent virus, there's selective pressure against that characteristic because you don't want to kill off the host. And so it's not clear why we should see an increased virulence emerging as D68 is evolving. And I'll give you some ideas about why we think that might be happening. So we can actually look at the evolution of the B clade, and based on the substitutions that we see accumulating, there were 530 substitutions that accumulated over an 11-year period here. And based on those substitutions, we can estimate that the evolutionary rate is 6.6 times 10 to the minus 3 substitutions per nucleotide per year. So this is an extremely rapid evolution. And over here in this kind of middle panel here, you can see we've kind of plotted where the evolutionary rate of D68 is in comparison to other kinds of viruses. And you can see it's really on the high end of the spectrum, very similar to the evolutionary rate that's been estimated for poliovirus, which is up here in this this point on the curve here, which kind of makes sense since they're both coronaviruses and some of these other viruses are DNA viruses, etc. Now, some of these substitutions result in non-synonymous amino acid changes. And predominantly those are found in the VP1 and VP2 structural proteins as well as in the 3D protease. So we decided to take a careful look at the substitutions in the VP1 capsid protein. And to make a long story short, here at the top row here are the different substitutions that we see in the VP1 capsid. And we look to see how they relate to what we know about the immune response in terms of antibody epitopes, which are, there are two antibody epitopes that have been well defined. One that neutralizes an infectivity of the virus, and that's indicated in this row here, the kind of the fourth row. And then this one here is a peptide that was bound by antibodies that were isolated from the CSF of AFM patients. And what you see is that in both cases, we see overlap between regions that are experiencing diversifying selection as the B clade has been evolving, and overlap with positions that are in these two antibody epitopes. And that suggests that the adaptive immune response, the antibody response, is driving the evolution of the virus. Now, what's really interesting is that in this middle row here, these are substitutions that have been found to influence the infectivity of the virus. So in this paper here by the Kupaveld group, they defined both sialic acid dependent and independent infection rate routes for the virus. And when they compared viruses that were capable of infecting without sialic acid, they found that they had differences in the sequence of VP1, and those differences were found at these positions here, 169 282 and 283. And both of those actually affect these antibody epitopes in diversifying regions of the virus. So in this slide, we've just mapped these positions on the structure of the capsid. And what you can see is that there is close proximity between the evolving sites in the B lineage, these neutralizing antibody epitopes, as well as these positions that are involved in sialic acid independent infection. And so what we think that this means is that, we think that the virus is evolving to evade herd immunity, and we're getting substitutions in antibody epitopes. But as a side effect of that, they are also affecting the receptor binding, and thereby possibly affecting viral severity by allowing the virus to change the cells that it's able to infect. And so what we think has been happening is that over time, the virus has been evolving to change its severity set point, which is kind of illustrated over here, from the non-B1 to the B1 to the B3 clades, where we've increased the severity set point in such a way that now we're seeing an increased rate of AFM development. So that's a very quick overview of kind of what we are seeing in terms of the evolution of D68, what might be driving the evolution, and how that relates to possible changes in virulence. And all of the work that I presented today was done by a graduate student in the lab, Yun Zhang. Thanks for your attention. And I want to then move right along to transfer the podium off to Dr. William Jackson from University of Maryland, who's going to continue the discussion of pathogenesis of D68 and its relationship with AFM.